You have to study humans, no models. You have to try and focus on the first cell, not the last cell. In other words, revolutionizing early detection. I'm going to hold this up. This is the cure for cancer.
Welcome to AI in Healthcare, formerly known as Target Cancer Podcast. My name is Dr. Sanjay Juneja. I'm a hematologist and medical oncologist. And here we are getting into all of the things on how technology and AI, at least as it relates to the specifics for what it can do for us in challenging disease processes, as well as in medicine, can be transformed and what to be optimistic about. Today, I have Dr. Azra Raza, who is an esteemed professor at Columbia University and is also an oncologist with a subspecialty understanding of everything related to the bone marrow and the things that have to do with MDS and leukemias, and one of the most challenging disease processes as a whole, in my personal opinion. But what we're going to talk about today is not so much what are these crazy new therapies and targets that we're doing, but really kind of rethinking how we can cure cancer. And by that, I mean in a capacity that we don't talk about enough. Because cancer, at the end of the day, especially when it comes to solid tumors, is usually a colony or a group of cells that have gone rogue. And if you can remove that from the body at a very early point, then the hope is that, you may not even need systemic drugs. So there are ways that we can find cancers and recognize cancers today. But the hope is, how can we find them even sooner? And that's what Dr. Azra is here to talk to us about. And Azra, one, welcome, and thank you for being here. And two, I'm very curious on what made you start recognizing this kind of like more global or macroscopic problem that we have.
Thank you for having me on your podcast, Sanjay. I've been a fan of yours, and the way you are highlighting some of the exciting news in AI and in medicine in general, I find it very alluring, let's say, because your canvas is so vast. Thank you for doing this.
Of course.
Look, I think that being an immigrant helped me, because I wanted to study cancer and treat the disease since I was a teenager. And I come to this country with this whole notion of curing cancer. And I was so anxious that I arrived here in January and my residency wasn't going to start until July. And I panicked thinking someone will cure cancer in these 6 months and I'm going to be left behind. That's how naive I was. So I went and started working as a fellow in oncology, actually, because I went and just offered myself and said, give me anything to do at the cancer center because I want to be involved. So I think being an outsider, being an immigrant helps you think a little differently.
100%. It's one opportunity, but really opportunity comes from, a more moral or ethically driven thing, which is just a need. And the need was that there is a debilitating illness and disease process out there that I know as an author you've written about, even on some of those elements that we can talk about hopefully on this episode. But when it relates to blood cancers, the therapies have really exploded. I hear that myeloma was one thing, that people still sometimes my patients fear when they hear, and I'm like, well, it's not, what it used to be. And then, of course, when it comes to pediatric blood cancers, the cure rate has just gone way up and everyone gets excited about CAR T, et cetera, which is why I thought it was very interesting that in our pre-call conversation, you had brought up, but Sanjay, what about the things we can do to before even hopefully one day needing all of those things? Can you characterize this kind of problem or uncovering of an issue that I think, I believe you get excited about, that can get some attention that maybe has been left behind because of tech and AI, or just scalable solutions that were not possible to be able to address really one of the earliest points and potentially the most impactful?
I think those are the burning questions of the time, actually. But going back to my own start in this enterprise, let's say, was that when I showed up at Roswell Park Cancer Center offering myself to do anything I could in cancer research and clinical cancer work, In fact, they had a position in pediatric oncology. That's where I began, but within 3 months, they basically threw me out. Not because I'm stupid, but because I was crying the whole time. I just couldn't sit on babies and do bone marrows and lumbar punctures on them. It was too painful. So they sent me off to adults. But you know what I realized within a very short period of arriving in the adult service was a defining moment where I realized that the same drugs that are curing ALL, acute lymphoblastic leukemia in children, which I had just been treating them with, have no effect in adults. Why? Because in a child's body, all the other normal cells are basically nascent and virgin cells, whereas in an adult, each cell has received 1,000 cuts by the time they're 70 years old. And so the way the cells metabolize the drugs is completely different in an adult as compared to a child. In other words, you could have the best solution, cure for cancer, which is working in children but not in adults. That also led me to realize the fact that even a littler version of our own selves cannot serve as a model for the adult body. In other words, all models by definition are fake. Because think of that surrealist painter, René Magritte, who has painted a picture of a perfect pipe, a pipe that you smoke, right? It's a perfect looking pipe. And the title of the picture is, "This is not a pipe." Why? Because it looks like a pipe. It's perfectly, anyone you show it to will say, why is it not a pipe? Because you can't lift it up and smoke it. That's why it's not a pipe. It's a picture of a pipe. And that's something in 1940 pointed out by the father of cybernetics, Norbert Wiener, who said that the best model for a cat is another cat, preferably the same one. In other words, the only model we can use is our own body, because the drug that works in me for the same cancer may not work in you because of your lifestyle, your metabolic pathways that have been shattered or that have been ignited. I don't know, but these kinds of things came as a sort of enlightenment for me. But if I had been raised in this system, I wouldn't even think about those things. You see, because it's like that frog, which is, if you throw a frog into boiling water, it's going to jump out. But if you keep it in a pot and start heating up the pot slowly, it will die, but it won't jump out because it gets desensitized by accommodating itself to the increasing temperature.
Whoa.
And so that's what I felt, that coming as an outsider, here is something. Number one, I realized that the same drugs won't work. And you know what I did, Sanjay? Before I published my book, The First Cell, because I knew it's going to be like the satanic verses of medicine. Everyone's going to hate me after I publish. One of the things I did is I went to 30 top leaders in oncology in the country, totally the top leaders, and asked them the same 5 questions. And one of the questions was just this, that if the same drugs are working in children but not in adults, what's your solution? It's such an important question. I just asked this a couple of years ago. Anyway, so the point I'm making is that you learn a few things, and I learned them very early on because I was an immigrant. Number one, that you cannot use models. I mean, if you can't use a child, forget using a mouse model. That is really idiotic. Number two, the important thing is, if you want to cure human cancer, you got to study human cells. You cannot make believe that you're taking human cells, putting them into an immunocompromised mouse in which you've killed its immune system, and then you want to give it you create something that you call cancer. But those are animals who don't spontaneously get those cancers. You're putting something in there, those cells completely change their nature. Then you treat them with some drugs you have concocted and they get cured. And now you bring those drugs to the bedside. Is it a wonder that 95% clinical trials today fail? And the 5% that succeed should have failed because they're only helping 30% patients for brief periods of time. So these are the things that affected me. Number one, the model system is bad. Number two is that you need to study human tissue. And number three, oh, by the way, so for the human tissue part, I started saving samples on my patients since 1984 when I realized this. To this day, I have collected 60,000 samples of blood, marrow, saliva, biopsies, you name it, serially obtained on patients as they go through natural history of the disease from pre-leukemia to leukemia or dying of end-stage MDS. This is such a precious tissue repository. No one else has such a thing. It's the oldest and the richest tissue repository. And do you know what, Sanjay? Not a single cell comes from another doctor. I've taken care of every patient myself. In fact, in my clinics twice a week, I do the marrows myself to this day. I'm working like an intern. Why? Because every vial in those 14 freezers has a poignant story for me. There is a patient behind that vial. And these are the things. So I collected all these samples. And the third important thing I realized is that the only good news we can ever give a cancer patient when we give them the diagnosis is, don't worry, we found it early. We can help you. If early detection is good, then why isn't the whole world studying and trying to revolutionize and focus on early detection? Why is everybody and their grandmother going after the last cell? It has never made any sense to me. And so I started my career studying and treating acute myeloid leukemia, but within a few years, I realized this is nonsense. AML is such a deadly disease that even God can't cure it, literally. Once it takes off, it's horrible. And I was right. Today, in 2025, we are using the same two drugs we were using in 1977 to treat this illness. 7 and 3, 7 days of RSC, 3 days of donomycin. What has changed? Nothing in practically 50 years. So I was right. I said to myself, the only way to help these patients is to find not when they're diagnosed with acute leukemia, but when they are at risk of developing acute leukemia. Is there such a population? Yes, pre-leukemia, myelodysplastic syndrome. And that's why I turned my attention to studying MDS. In other words, my mantra all my life, since I'm 28 years old, has been, number one, you have to study humans, no models. Number two, you have to try and focus on the first cell, not the last cell. In other words, revolutionizing early detection. Why is it so important? Because not just cancer, but every disease is a silent killer. Our bodies are so well equipped with repair toolkits that they keep compensating and compensating in an organ until 90% of it is gone. They don't send any warning signs that a leak has developed. Until the roof caves in, you don't know. Total dementia appears, then you know that, oh, something had been going on for 20 years to lead up to this. And the same thing. So in other words, if there is a silent enemy sitting in us and we know that for the first 50, 60 years we can handle it, but then everything is going to go crazy after we turn 60 or 50, well, then why aren't we preparing to detect these things early, to detect the whispers before the scream comes out?
To unpack it, because there's so much to unpack. Number one, I loved what you said at the beginning, which was the way, and I think, there was celestial ordination on why you went from pediatric to adults, because it gave you this realization in the same way that when we are the newborn, cute little baby and before we're walking, we haven't developed the calluses on the way we preference to walk on the bottoms of our feet. We haven't then over decades gotten the arthritic or degenerative changes in those points that basically in your knee and hip may relate to where you prefer on your foot. And on top of that, if anyone, first of all, that's going to change anyway. So that's your point, is like, how are you going to, you can maybe have a brace or a technology or a device before someone starts walking when everything is relatively, what I will say is germinal. But you can't have a general, applicable brace or foot insert or anything mechanical 60 years in, because it's all going to be varied. And you're saying the same thing has to do with a cellular level on the metabolics and how this stuff is processed and not processed and what escape mechanisms. Those kind of things happen over your lifetime. And you're like, how are we trying to put, it from one place to another, and that it doesn't make, a lot of sense to you. And that is the way that we're doing it. And how we're extrapolating it is, take this 65-year-old, what kind of brace do they need? And it's like, I don't know. I need to know, basically, what was their mechanical problem, where, then I can get an idea of where their degenerative disease process is, not even including the fact that we all have polymorphisms and inherited properties that may or may not predispose us to all that. Now, with all that said, the point that you're making about before the problem, I still remember that I had a staff when I was in residency that said, Sanjay, look at the bottom of the shoes. And I said, why? Because they were complaining about the hip. And where he showed me the erosion was from friction, he was like, he has a leg length discrepancy. And I said, what? And he says, one's longer than the other. And we went in there, and sure enough, one was slightly shorter than the other. Well, at this point, he's got this debilitating compensatory, process over several decades, that at the end of the day, if he had an earlier, recognition and changed to his leg length discrepancy, all of the things that went downstream and upstream in his spine and his hips and further down and all the mechanical issues could have been prevented, much like cancer and the upstream and downstream issues. And in case somebody's listening and can't appreciate, AML, and correct me if I'm wrong if it's all the time, but when you have myelodysplastic syndrome, so a process in the bone marrow where it's not something you see in young kids, but over time it just kind of gets, like a good garden that you buy in the house, a new house looks great, the weeds start coming out and all these other, kind of invasive species take away from the ingredients, that can turn into AML. So you're like, I've treated AML well. I want to talk about the stuff that we know that some AMLs come out of. And that's what focused on this pre-disease state, awareness or disease process you're talking about. Now, with all of that to kind of set the landscape, where I get super excited about what you're about to tell us is, what I think you're going to tell us is this longitudinal data collection that you've had. Meaning, it's like we get labs year to year, which many argue need to be more often, which is why they have, insulin devices for diabetics, so they can get a better idea than just, a shot in the dark. You're saying you have collected serially and continuously all of these different states for people that ended up with leukemia, didn't end up with leukemia, had MDS, stated MDS, didn't have MDS, and I assume identified because the kind of properties and constituents and other factors that existed in this specific person. And you just listed 60,000, either samples or patients, probably samples. With all of that said, I think what you're going to get to next is how you can see and appreciate AI and its basic principles of being able to do many, many things in parallel, or larger things, but in parallel to an extraordinary amount of consideration more than the human mind. And I assume, give us some examples of how this kind of continuous collection can really uncover and dig out and surface insights only if you have the pre, post, and everything in between. Like, that's the way that you can apply machine learning and uncover, basically patterns, where AI is, at its most traditional sense, pattern recognition is the simplest version of AI, to be able to ascertain those things. So tell me if I'm wrong or right, because I'm all ears.
You're completely right, of course. Look, right now, the only way we are detecting early cancer is monitoring or screening for 5 common cancers, PSA for prostate, pap smears for cervical, you do colonoscopy for colon, you do mammography for breast cancer, and you do CT scans of the chest for people at high risk for lung cancer. So just to put things into perspective, we detect 9 million positive tests out of these 5 screening measures. But a positive mammogram doesn't mean there's necessarily cancer. It could be a cyst. So then you go in and make an actual pathologic diagnosis, and out of 9 million, 8.8 million are false positives. So we are spending in this country $34 billion a year to detect 204,000 cancers early. Those are the real statistics. Nobody wants to think about it. It galls me to think that in this day and age of predictive analytics of AI and Fitbits and Aura rings and Apple watches and all these things, we are actually depending on a GI person to go in once every 2-3 years into the gut and look for a tumor by colonoscopy. That really is so counterintuitive that it's shocking when you really sit back and start thinking about things. The problem, Sanjay, is we don't see things as they are. We see things as we are. Because I have been trained in this way to look at things, that's the only way everything will appear to me. And I keep saying that being an outsider helps me all the time. So here is my solution for all of this. I'm going to hold this up. This is the cure for cancer. But the cure is not in the syringe. The cure is in the needle. What is in this needle? In this needle is a stent. Like a stent that is made of cellophane or something very thin film, just like a coronary stent or your stent you put in the kidneys or in the bile duct. This contains a stent, which using the syringe can be injected into a blood vessel. It goes there, unfurls itself and sits there forever. And what does it have? It has these electronic circuitry on it and a built-in little chip. The electronic circuitry will produce a current in the blood vessel so that anything which passes through now causes impedance of the current, which is picked up and recorded on the little chip in the computer that's right on this inside that stent. Okay, this is not a pie in the sky. This is not something I've imagined. I'm holding this up because biomedical engineers Sam Sear and Ken Shepard at Columbia University, with whom I've been working for 10 years, have actually made this stent. We call it the Sentinel, combination of a stent and sentinel. And the idea is that this kind of an implantable device can be inserted at birth and can continuously monitor for the appearance of abnormal cells, because cancer cells are large. If it detects a large cell, there'll be a larger electronic signal, and your app on the phone will get the message. And then with machine learning, these things are going to get so much better with time that, even that it will be able to tell the difference between what's real and what's not very quickly. It's already happening as we insert these kinds of stents into pigs. But let me just give you one example. Grail is a company that is dedicated to early cancer detection. And the way they're doing it is through liquid biopsies, even to find solid cancers, which means, how do you do that? Well, a solid cancer, let's say a cancer in the pancreas or in the liver or lungs or breast, the cells of that cancer, as they die, will shed pieces of DNA into the blood. And in the beginning, people started to note that if that tiny piece of DNA has a mutation that is associated with pancreatic cancer, then that means the person could be having a pancreatic cancer. But the way Grail started to train its AI platform by feeding it gazillions of data bytes, you know what started happening now, Sanjay? That the machine learning is so supreme that a mutation is not even needed anymore. The AI platform has trained itself to simply look at a tiny piece of DNA and say, hey, this is coming from a normal cell, this is coming from a malignant cell. You have no idea what the machine has learned, on what basis is it doing. Is it on the basis of the number of methyl markers, methylation sites that it is deciding? But now they don't care about finding a mutated piece of DNA. So I'm saying to you that the same way, actually using the current technologies, we can do so much better by trying to find cancer at not stage 1. Even Grail is trying to find earliest is stage 1. I'm saying no. Stage 1 is too late. Why? Because you already need those Paleolithic cavemen kind of measures of slash, poison, and burn. Those things are not patient-friendly or compassionate treatments. And let's stop giving ourselves gold medals for using those on poor patients. It's just the most horrendous thing you can do to people. It's like taking a baseball bat and hitting a dog with it to get rid of its fleas. That's how horrible these treatments are. But even stage 1 requires one or the other. So my point is, stage 1 is too late. I want to find the first cell stage. What is the first cell stage? That's very different than stage 1. A stage is set because of chronic inflammation, toxins, infections, whatever is going on in some organ. A stage is set for the first cell to arise. We want to detect cancer even prior to the first cell, at that first cell stage. And that is only possible by continuous monitoring, by treating the human body as a machine and screening it 24/7 from birth to death.
You're so right. And when you're saying the first cell, you don't even mean the first invasive cell. You mean just the first cell in and of itself. So what I'm thinking about is in situ, right? So that's kind of a place of confusion for people. Like with breast cancer, it's stage 0, and then tuberculous adenoma, which is a pre-cancer, potentially cancer lesion. That's a polyp in the colon. So you're not even just talking about a cancer cell that hasn't become invasive yet. You're talking about even a pre-cancer cell state that is still a cell and hasn't even gotten the potential or the constituents to be able to become invasive disease. And then secondly, when you're talking about finding that pre-cancer state, because it's an evolution, for the most part, these things aren't a switch. It's not like it's all good and great one day and all of a sudden went rogue. There is some period of time, which kind of scares me about things, in other climates outside of healthcare, is where this transitional phase happens and it just becomes kind of the way that you talked about the frog in the pot. It's like you don't even know that there's a threat because you're dynamically basically calibrating yourself or adapting. There is that same dynamic state with cancer cells, and it is not a flip of a switch. And to your point of these, millions of screening that we have, we're just taking a picture of a moment in time and very seldomly actually comparing it side by side. But the thing that I get very excited about is when you're talking about how we don't even know how or why it's telling us that that's the case. All it knows is this thing that it's finding seems to be very well affiliated and correlated with a cancer down the line because it's been fed the data. It kind of reminds me of something called famously Move 37, which that happened in, I believe, 2016, where AlphaGo, which is an AI,
I know that very well.
right? Like
game
model that played Go really well. And it was against the grandmaster, the biggest grandmaster, right, in the world in Go. And all of the room was filled with a bunch of Go specialists and grandmasters. And I love this example that I sprinkle my own anecdote, is because these grandmasters didn't just come out of the blue oftentimes. A lot of times they were taught and handed down generationally by their parents and everything. That's how you get really good as a grandmaster, especially with something as old as Go. So even with all of that, on Move 37, AlphaGo made a move where everybody audibly gasped, and it was like horrified, like, or like, in shock. Couldn't believe that AlphaGo did something that made no sense to a room full of grandmasters and their generational learning. turned out it won. And it didn't take long to win after that. But it was a move that was just inconceivable to the own completely unaware biases either that we have, or not even bias, but the mind doesn't know what the eyes haven't seen. And this whole concept of a fresh set of eyes or new, you just can't remove it from human experience. And that is where I get super excited, and where you're telling us you get excited, because it's like, let's stop spending millions and billions of dollars in our own, what does Carl Sagan say? The folly of our human conceits, and thinking we're going to crack this code and we're going to come up with an idea and then test in a model. Let's just look at what is already happening, because there ain't no question that this stuff is happening. And we have plenty of data and sites and samples in front of us. But let's be humble enough to try to figure out what some of those things are, rather than trying to figure them out within the, limitations of one human mind. So that entire story that you just kind of gave us with what the capabilities of this device are, and again, then not even knowing why, we just know that relationship exists. Can you give us, I guess, even more of an idea of what we can do with that, what it'll disclose, and hopefully what is necessary to get on board with this? Is it pride? Is it protocol? Is it structure? I mean, what can we do with it? Because I think that's what ultimately everyone wants, just like we had gotten polio rid of and measles and some of these other things. What is one thing that we could get rid of cancer by? Just not ever letting it be. I mean, that's what we achieved with other diseases.
Thank you, Sanjay. Well, you said so many interesting things. First of all, I loved that documentary of AlphaGo. I watched it many times, and Demis Hassibis obviously got the Nobel Prize as a result of just exactly starting that. That's where he got his go. And I think that it's a very important example that you just gave, that the fact that we have ourselves created a machine that can teach itself to think 70 steps ahead. Humans can only think 2-3 steps ahead, but that thing can, if our IQ is the highest recorded is 176, the IQ of a regular AI thing is 2000, and that of super ones is going to go into millions. So how can we compete? But we have created these machines that can do this stuff for us. So, for example, I'll keep coming back to this, to the device, to say that it doesn't just detect cells. It can detect biomarkers such as what the cells are shedding, pieces of DNA, pieces of RNA, pieces of protein. So peptides can be detected by using single molecular electron field transistors, can detect all these biomarkers. But let's say it starts, you see, the difference is that Grail is using AI right now to enhance their machine learning capabilities, and I just told you that they've become so much better. But their problem is they're still dependent on periodic testing, once a year or twice a year at most. But what I'm saying is that while you're waiting for that 1 year or 6 months, you can develop a cancer that then requires slash, poison, or burn. So what you need for continuous monitoring, this device that I just showed you can sample the entire 5 liters of blood for all these biomarkers and any abnormal cell within 18 days. The entire body's everything will be. So imagine this thing is sitting in your blood vessel, constantly monitoring. Every 18 days, every bit of your blood has been monitored or has been screened for any abnormality. And I'm saying that these are the kinds of implantable devices that are going to have to replace all kinds of disease detection. Because what we are aiming for is to detect that wellness to illness transition, even before the transition point actually happens, just the whisper of it. And at that point, fixing things will be so much easier. You could probably have a health drink which is supplemented with the exact right kind of antidotes that you need, or a simple lifestyle change could make a difference at that time. So I feel that having the kind of technology that's available, and I've been a miser in one sense, although I've published hundreds of papers using so many of my samples, but I only use samples where I have duplicates available. Because I've been saving samples for the one grand study we'll be able to do. Now technology has arrived. So now I can take serial samples. Some people who lived for 10 years, some who died within 6 months. I have serial samples on both. But the most important is the pre-diagnosis sample on each. Could I have predicted that this one is going to survive 10 years and this one is going to die within 6 months? And if yes, what do I need to do with it? Well, right now what we can do is perform multi-omics, which is, genomics, transcriptomics, metabolomics, proteomics, multi-omics on these samples. And we figure out that this is the combination of signatures that tells us that things are going to move aggressively in this person. If we have that kind of a signature, even before this implantable device gets approved by the FDA, we can start using those kinds of signatures not once a year or twice a year, but with point of care technology. You know what that is? That is that, again, biomedical engineers, Professor Sam Sia at Columbia has built literally a machine that's the size of a toaster, which can perform PCR, RT-PCR, all kinds of things on a drop of blood. So sitting at home with this little cassette that you put into this toaster-like machine, you can test yourself every week, every day, every month, whatever you want for this signature while we are waiting for the implantable devices to be approved. I'm saying the problem is, Sanjay, no one wants to think like that. No one wants to think of, I am desperately trying to raise resources and funds to help me and my colleagues, the biomedical engineers, to get this thing to the bedside. We are saying that given my tissue repository, well, basically, they have developed this device. But what should the device detect? I have that. Cells and the biomarkers. That's why our complementary strengths are so important. Do you know we are claiming that within 6 to 8 months, we can have all the biomarkers identified because we have all the sample. I just have to mail it or just take it down to the Columbia's core facility for doing the multi-omics, and they'll give me within 3 months. And then I spend 3 months analyzing the data to come up with the signature. And they just need to finalize something points in their device so they can start putting it in large mammals. Given the right resources, we can do this in 6 to 8 months, but no one is willing to give the resources. I have been groveling in front of billionaires, in front of big pharma company, in front of academic leaders, everybody. No one wants to listen to me because all the money is in the other aspect, the last cell and the treatment developments. So you tell me, Sanjay, you interview a lot of people. Who do I go to? Who needs to listen to me?
I've had to experience the very quick disillusionment and harsh reality of healthcare in this country is just different than it is elsewhere. Like you don't make progress and productivity, one, by not knowing it, two, by acknowledging and harping on it, but three, the way you do it is to accept it and thread the needle, which I know you know well, and unfortunately it sounds like have laboriously appreciated. I think one place that we could actually really kind of find a proof of concept, and before I say this, I need to orient for anyone listening. When y'all hear biomarkers, and when you're hearing about this signaling on the before space, I actually just watched a Netflix show, documentary called Inside the Mind of a Dog or something like that. And apparently dogs, when they smell in 3D. So like where we can tell when we hear, where something is, an idea, even with your eyes closed, on is it in front of you, behind, but to the left, to the right, in 360 degrees. They can apparently do that with smell. And that made me immediately think back to when I first was told that dogs can predict seizures and they can actually predict cancers potentially, or death. And I was like, that's nonsense. There's no way. They're not like, spiritual or on some, different plane of time. But really what the science is showing is they're able to smell the byproducts of this very complicated but highly reproducible and orchestrated system that is the human physiology and biology. So like on the skin, the discharge elements, electric stuff, the catecholamine releases and what that does, all of this is so beautifully harmonious and dynamic that if it can do it by smell, you're talking about something in that same capacity when it comes to environmental orientation, landscape signaling, the same way a fire is there with the smoke. You don't have to see the fire, but the smoke is there, and all of these things, the way we can predict wildfires and everything. You're saying, Sanjay, and if anyone doesn't know this, all of this circulates. The blood is literally the room with which sound travels. It's the same thing for everything in your body. Your entire blood system goes through every single cell in your body. I mean, that's a fact. This is the highway. And so you're saying, let's analyze the highway for something that flows into every cell that we have and see when the callus formation is happening on the foot compared to like the baby soft foot, or when all of these things happen when it comes to these changes before it's too late, like the leg discrepancy. All of that to say, what I'm thinking is, instead of nixing it forever, one area could be potentially where you have patients that have been cured of cancers. So you already have a head start on what changes dynamically happened, if you're lucky, if you had enough samples that it just didn't walk in and one day are diagnosed, because then it's useless, right? Except for what you have today. But if you have a serial idea and AI can say what mutations likely occurred over what period of time to eventually, quote unquote, get here, get unregulated, which is what cancer means. And then you apply this in these stage 2s or stage 3s, not necessarily for recurrence even, but even the ones where we know second primaries happen, which that means that there are cells that are along the process of becoming a cancer, whether invasive or not invasive, but there's steps along the way, meaning tubulovillous adenoma versus, something that's less evolved. Now you have a narrowed scope of the 2 or 3 steps remaining before it becomes cancer. So it's not necessarily for the recurrence of the same previous one, even though I could conceive an area where that makes sense. But the second primaries, the colon, the lung where smokers happen, like lung cancer patients get a different one because they've already had a few of those steps, in the area and in the colon. The rest of the conversation you and I are going to have offline. But certainly I think these are ways that one can thread the needle, because you can have a therapeutic intervention and quickly, because you're able to say, aha, the same thing that made it HER2 positive, or the same thing that made it, whatever the case may be. There's still that economic sad reality. But 2, it will give us an idea of then hopefully how to avoid those second primaries, which will be preventative. I don't know. That's me thinking out loud and in real time. But I just hope that by listening to you and your insights and your collective, aggregate of observation and now fervent need for action, that anyone can appreciate this is a dynamic evolutionary process. We don't get enough pictures of the evolution. We have a gazillion sad examples, the aggregate that is the tragedy of cancer and suffering. We have all of that out there, and we're not doing anything to serially monitor it, because we know that we have clues that happen before we get there. We know that. And yet we're like, meh, we'll wait till we get there. And that's maddening. So, whereas a lot of these, podcasts will have like what needs to happen in a technical aspect or regulatory or whatever. This one's scary, but so large in its return is, how do we get people on board with what we know to be true? And that is the number of steps you have to get to third base before you get to home base in baseball. There is no rule where you're going to get to first and then home. That's just not possible. And the same things for cancer, and yet here we are.
Well said. Let me add a couple of things to it. The first is that I think everybody who has any sense realizes that the future of all of healthcare is in prevention, not reactive, but proactive care. And that depends on early detection. That's the only way you're going to be able to prevent things, by finding signs of early detection. Number two, as you said, we need ecosystem integration. We need an understanding across the fields and across disciplines to accept this, and at least for the future, try to work towards that. Number three, you made a reference to looking at people who are at risk of second malignancies. You know that 6 years ago, I created a think tank, the oncology think tank, where I invited 30 top leaders in the country, Harvard, Yale, Stanford, Hopkins, Columbia, top oncology leaders, and industry, Regeneron, Grail, Bruker, Patrick Soon-Shiong. I held 17 two-hour-long meetings with all these people. And then we published an opinion paper in Scientific American. I'm the lead author with 30 co-authors trailing me from all these institutions. And the consensus, because you see, art is I. Van Gogh can stand and make the Starry Night by himself. But science is we. As I said, ecosystem integration and consensus building is key to success in science. Right? So we held 17 two-hour-long meetings debating every aspect of cancer, and I pushed everybody to either accept that early detection is the right answer or point out the fatal flaw to me. And they couldn't, so we all agreed, and we wrote the consensus paper that the future is to detect at the first cell stage, not stage 1. So now I have this paper that was published in 2021 with all these colleagues saying the same thing. And we identified cancer survivors as a high-risk group. Because do you know, Sanjay, that 20% of all new cancers in the United States, 1 in 5 occur in a cancer survivor. Now, I don't want anyone to get anxious on this who's listening to the podcast. Maybe you have had cancer. Maybe someone you love is a cancer survivor. For an individual, the risk is only slightly higher, 13% higher. But if you look at the actual numbers when newly diagnosed cases, then if there's 2 million new cases of cancer this year in the US, 400,000 will be in a cancer survivor. So do you know what I started doing 6 years ago? Well, 2015, 10 years ago, I started to filter the blood of all my patients, trying to catch the first cell, catch the first biomarkers. And lo and behold, we started catching these giant cells. I've been publishing on it extensively. But what happened is that I went to establish a tissue repository of cancer survivors and started just filtering and saving the blood of all these cancer survivors coming to Columbia University. And now I have a tissue repository on cancer survivors. And many of them have developed a second cancer, new primaries, as you say, new second primaries, not a recurrence. Can you imagine? I have the samples from before and after. But where is the will to study? I mean, I need help. I need other people to recognize the urgency, because I'm the one seeing 30 to 40 cancer patients every week in my clinic for the last 40 years. And I'm the one giving bad news after bad news to these people. Something that Henry Ford said in 1909 made a big impact on me when I came to this country. He said, Sanjay, that if I had asked my customers what do they want, they would have said a faster horse. Instead, I gave them an automobile. In other words, if you ask the customer what they want, or if the customer tells you what they want, it's already too late. You've lost the game. I am saying that this is not just an automobile in the time of horse carriages. This is a supersonic Concorde. This is the ultimate. This is what we need. AI-driven implantable devices to continuously monitor 24/7, from birth to death, from womb to tomb, from lust to dust. Every time, keep sensing for the first signs of disease. And nothing is better than blood serving as a window into all these transitions. And that we can do it. The will has to be there. Somebody has to have the clarity of vision to step up and say, yes, what you're saying is right, Azra. Go ahead and do it. Instead, everyone is putting roadblocks on my way for the last 35 years.
I'm going to help it. We're going to make it happen. We're going to connect offline of this. And I'm telling you, there are people that share your sentiment. There are people that are just hungry and are like, hey, I just need, anyone to tell me how I can use the resources. So we're going to make that happen. There's a lot of conversation to follow up here. I promise you that. But for the sake of the podcast, I'm going to say real quick. So you mentioned, kind of these clues, and you even said it's unpopular, and that's why, I've had kind of Dr. Jason Fung, Dr. Thomas Seyfried, really talk about this cancer process as a mitochondrial injury or something to do with metabolism. But the point is, we don't know what we don't know, and we should definitely look and explore. We had a good, apparently, friend of yours and mutual friend is probably the most intimidating person I ever talked to just because of my sheer ignorance on the subject. But Michael Levin is now introducing this whole concept of bioelectric escape and all these things that actually happen when the harmony of the bioelectric existence that is disrupted, and all of a sudden something is just evolving in an autonomous manner because it feels like it has to back in the primordial soup days. So there's all kinds of stuff that we can look into. And then also, to your point about endoscopies, we had a good podcast with Dr. Michael Byrne where, AI is actually being applied to these scopes so that you can actually in real time, be able to recognize things that may look different with someone with a curry diet and turmeric because their intestines look different than somebody that's, predominantly meat and potatoes. And so there's a long way to go, but there's no question that we have all of the clues, if not even beyond clues, all of the realities of why and when things become cancer. And your argument or your position, it's not an argument, is why aren't we looking at it? So with that said, Dr. Azra, I think we'll have to have you back, hopefully on a good follow-up call based on where we got after this podcast and with this device and the applications that we have.
Look, Sanjay, the one thing I want to impress upon everybody is my sole motivation is not any personal gain or glory. Rather, it is the patient's pain and anguish. And the book I wrote, The First Cell, the subtitle is "And the Human Costs of Pursuing Cancer to the Last." Because in my book, what I've done is basically I have looked at everything to do with cancer from the perspective of human pain and suffering. What is it we are doing to patients? And I don't know anyone else who did this, but I went back 10 years after people had lost their loved ones who were my patients. I went back to the families 10 years later. And now I ask them that with the luxury of time and the benefit of perspective, and that chaotic time you lived during which your loved one was sick and treated and dying when you couldn't think of anything else but what you had to do at the moment, with this perspective, would you change any of your decisions? Would you do things differently? And the last part of my book is all dedicated to the answers that the families give about the patients I talked about. So I have 7 chapters in the book is dedicated to a patient. And I go to their families, and then, do you know how sad the situation is that not one person said they would change anything? Why? Because they said, what were the alternatives? The only alternative they were given were either your beloved one is going to die of cancer or the treatment. So, which is more painful? Dying of cancer is more painful. Think that the outcome for stage 4 glioblastoma, stage 4 pancreatic, stage 4 of practically any cancer you take is no different than it was 100 years ago. So yes, we are curing 60% patients, but with slash, poison and burn because they were diagnosed early enough. 40% we haven't touched them basically. Except anecdotal things with CAR T, we have treated a few thousand, 27,000 people in 10 years. What is that compared to 20 million who were diagnosed with cancer? So let me just end by saying that it's all about the human suffering and anguish. Emily Dickinson. I measure every grief I meet with analytic eyes. I wonder if it weighs like mine or has a different size.
Unreal.
I wonder if they had it long or did it just begin. I cannot find the date of mine. It's been so long a pain. I wonder if it hurts to live and if they have to try and whether could they choose between, they would not rather die.
I have chills. I have goosebumps, especially with the applicability of that statement even beyond. I mean, that's heavy.
This is what you and I are dealing with in oncology.
Yeah.
They would not rather die if they had a choice. These are the choices we are giving them. How dare we give ourselves gold medals and prance on stages of international meetings congratulating each other? When is the last time we thought about all the people who are dying in our hands instead of presenting that positive anecdote who survived?
Because it's such a deep aggregate of tragedy. Like it's like if for anyone that thinks success, it's like it's tragedy. It's tenfold more than the success element.
To begin the ending, you have to end the beginning.
That's the quote. That's what my notion's going to be. Golly. Thank you.